common drugs of chemotherapy covers anti-metabolic drugs, antibiotics, and the traditional Chinese medicine effect components. Antimetabolite is a compound that blocks metabolic pathway function by competitively inhibiting a key enzyme because it closely resembles the normal enzyme substrate. Sulfonamides or sulfa drugs are structurally related to sulfonamide, an analog of p-aminobenzoic acid or PABA. PABA is needed for folic acid synthesis. Folic acid or folate is a precursor of purine and pyrimidines, the basis used in the construction of DNA, RNA, and other important cell constituents. When sulfonamide or other sulfonamide enters a bacterial cell, it competes with PABA for the active site of an enzyme involved in folic acid synthesis, causing a decline in folate concentration, which indirectly impairs bacterial growth. Since Fleming's discovery of penicillin, many antibiotics have been discovered. However, most antibiotics fall into a limited number of classes. Part of the challenge in developing new antibiotics is to fight bacterial structure or process not already targeted. Here we present some of the more common antibiotics according to their functional and structural class. Firstly, inhibition of cell wall synthesis. The most selective antibiotics are those that interfere with bacterial cell wall synthesis. Drugs such as penicillins, cephalosporins, vancomycin, and bacitrin have a high therapeutic index because they target structures and functions not found in eukaryotic cells. Secondly, inhibition of protein synthesis. Many antibiotics such as chloramphenicol, erythromycin, and streptomycin inhibit protein synthesis by binding with the bacterial ribosome and other components of protein synthesis. Because these drugs discriminate between bacterial and eukaryotic ribosomes, their therapeutic index is fairly high but not as high as that of cell wall synthesis inhibitors. Thirdly, inhibition of nucleic acid replication and transcription. Quinolones or rafeprin inhibits DNA polymerase and topoisomerase or RNA polymerase to block replication or transcription, respectively. Fourthly, drugs like polymyxin B causes injury to plasma membrane. Last but not the least, several valuable drugs act as antimetabolites. For example, sulfonamide or tryptophan prime analogs or block the functioning of metabolic pathways. Even antimicrobial drugs target certain essential functions of the microbe. It must not interfere with essential functions of the microbe's host. This table shows the spectrum of activity of antibiotics and other antibio drugs. For example, isoniazid is only effective for treating mycobacteria. Penicillin G is used for gram-positive bacteria. By contrast, streptomycin and tetracycline have a relatively wide range of antimicrobial effects. Ketoconazole are used for fungi treatment. For malaria or tapeworms, mefloquine and niclosamide are being used. Acyclovir is a commonly used antiviral drug for treatment of herpes infections. Most penicillins, like penicillin G or benzopenicillin, are derivatives of 6-aminopenicillinic acid and differ from one another with respect to the side chain attached to the amino group. The most crucial feature of the molecule is the beta-lactam ring, which is essential for bioactivity. Many penicillin-resistant bacteria produce penicillinase, also called beta-lactamase, enzymes that inactivate the antibiotic by hydrolyzing a bond in the beta-lactam ring. The structure of the penicillins resembles the terminal D-aniline found on the peptide side chain of the peptidoglycan subunit. It is thought that this structure similarity blocks the enzyme catalyzing the transpeptidation reaction, forms the peptidoglycan cross-links. Thus, formation of a complete cell wall is blocked, leading to osmotic lysis. This mechanism is consistent with the observation that penicillins act only on growing bacteria that are synthesizing new peptidoglycan. Here you can see in this figure, the bacterial cell lysing as penicillins inhibit the bacterial cell synthesis. Here is a table listing some antibacterial drugs, especially inhibiting bacterial cell wall synthesis. Penicillin is used against gram-positive bacteria, while some semi-synthetic penicillins have broad spectrum. Aztreonam are effective against gram-negative bacteria. First generation of acts similar to penicillin with the development of theory and technology. The fifth generation of cephalosporins can act against MRSA, the methicillin-resistant Staphylococcus aureus. Here are some examples of other kinds of antibacterial drugs we have mentioned before. Inhibitors of protein synthesis, such as aminoglycosides, tetracyclines, and glycosylcyclines usually have a broad spectrum. Erythromycin in macrolides is an alternative to penicillin. Polymyxin B is one of the lipopeptides used to treat MRSA infections. Rifapentine is a nucleic acid synthesis inhibitor used in the treatment of tuberculosis. Then the antifungal, antiviral, antiprotozoal drugs and their usage listed. Determination of antimicrobial effectiveness against specific pathogens is essential for proper therapy. Testing can show which agents are most effective against a pathogen and giving an estimate of the proper therapeutic dose. Dilution susceptibility test can be used to determine minimum inhibitory concentration, MIC, and minimum lethal concentration, MLC, values. Antibiotic dilution test can be done in both agar and broth. If a rapid growing microbe such as gram-positive Staphylococcus or the gram-negative Pseudomonas is being tested, a disk diffusion technique may be used to save time and media. The principle behind this assay is fairly simple. When a different antibiotic impregnated paper disc is placed on agar previously inoculated with the taste bacteria, the antibiotic diffuses radially outwards through the agar, producing an antibiotic concentration gradient, as shown on the left figure. However, the e-test may be used in sensitivity testing under a majority of conditions. It is particularly convenient for use with anaerobic pathogens that grow relatively poorly in broth culture but quite well on agar. In general, bacteria to be tested for antimicrobial sensitivities are individually inoculated on the entire surface of an agar medium, and the e-test strips are placed on the surface so that they extend out radially from the center. Each strip contains a gradient of an antibiotic and is labeled with the scale of MIC values. After 24 to 48 hours of incubation, an elliptical zone of inhibition appears. MICs are determined from the point of intersections between the inhibition zone and the strip scale of MIC values. In the broth dilution test, a series of broth tubes containing antibiotic concentrations is inoculated with the standard density of the test organism. The lowest concentration of the antibiotic resulting in no growth after 14 to 20 hours of incubation is the MIC. The MLC can be ascertained if the tubes showing no growth and then cultured into fresh media lacking antibiotic. In other words, the lowest antibiotic concentration from which the microorganisms do not subsequently grow is the MLC.